We're gonna get started right away with our next session. I'm Joshua Schimmer from the Piper Jaffray biotech team. Pleased to introduce the Chief Financial Officer of uniQure, Matt Kapusta. uniQure is, I think, hopefully everyone knows, a pioneer and trailblazer in the field of gene therapy. So, Matt, why don't you give us a snapshot of the company and what you think differentiates uniQure from some of the other gene therapy players?
Sure, Josh. Great to see you as always. Thanks for attending the fireside chat here. So I think there's a number of factors that really differentiate uniQure. I think the first one that we talk a lot about, and unfortunately maybe one of those that I think is undervalued today, is the manufacturing capabilities. And uniQure made a strategic decision a number of years ago to invest heavily in owning and controlling its manufacturing capabilities. And when you think about cell and regenerative therapy, in particular gene therapy, the process is the product. And so we have the only EMA-approved gene therapy facility in Europe. We currently manufacture Glybera out of that facility, and it's responsible for early stage development. We also invested a significant amount of money in commercial grade and late stage manufacturing capability in Lexington, Massachusetts, right outside of Boston, where I'm currently based and our CEO is currently based. That facility has the ability to scale up to 2,000 liters capacity, and we spent 10 years really perfecting the manufacturing process. That's assay development, process development, stoichiometry, the relationship between each of the different viral vectors and genes. So we're very thrilled to have that capability. We think it will be extremely important in the future as we get into late stage studies and commercialization. I think the second thing that is differentiating to us is our proprietary AAV5 vector. I think a lot of people aren't aware that, but we actually have 3 human clinical studies in over 20 patients of experience with AAV5 in both the liver and the brain. The safety profile is, we think, quite differentiated. We've dosed up to 2E to the 13 in acute intermittent porphyria without any safety issues. And we think we have clinical proof of concept in both the brain with Sanfilippo B as well as the liver with some of our early and encouraging data in Hemophilia B. I think the third element of differentiation is our broad clinical pipeline. We think we're one of the few gene therapy companies that actually has 3 clinical studies that are ongoing. So we have 1 phase 1/2 study in Hemophilia B. We've completed a phase 1/2 study in Sanfilippo B and have an ongoing phase 1/2 study in Parkinson's disease. We also have an interesting early stage pipeline. In fact, we'll be announcing today proof of concept in Huntington's disease, a publication that'll be coming out today, where we've looked at in vivo and in vitro in a humanized mouse model and rat model, the successful knockdown of the Huntingtin protein, and we'll be taking that forward into IND-enabling studies and hopefully into the clinic next year. And then I think the last point of differentiation, Josh, I think is in our Hemophilia B program. There's a lot of folks going after Hemophilia B, but I think we're the only ones that are using the same exact wild-type gene cassette that now has 5 years plus of durability that's been proven in the Professor Nadvani St. Jude's Children's Hospital study that was published in the New England Journal of Medicine. So we're very much looking forward to that, pardon me, and hopefully exploiting those areas of differentiation going forward.
Well, great. A lot to work with. Maybe we'll start by focusing in on the Hemophilia B program, because that's certainly been an area that I get a lot of investor interest regarding. And just take us through what you've shown so far, where the program is, and what the next steps that we can look forward to are.
Sure. So as I mentioned, we have an ongoing phase 1/2 clinical study in Hemophilia B. We currently have 9 sites in Europe that are enrolling patients. We've completed the enrollment of the first cohort, which included 5 patients and was dosing at 5 times E to the 12th. We announced early and encouraging data in early January that suggested that the first 2 patients that had crossed the 12-week post-follow-up treatment point had achieved factor IX expression of 4.5% and 5.5% respectively. All of these patients in this cohort had less than 1% factor IX endogenous expression levels. So that was very encouraging. We also communicated that 4 of those 5 patients were off of prophylaxis therapy. And that's very important because that is one of the goals that we're trying to do in this study. We're trying to take these patients that are on once, twice, often 3 times weekly infusions of prophylactic enzyme replacement therapy and shift their phenotype from severe hemophiliac to mild hemophiliac, which means they would no longer be susceptible to spontaneous bleeds and no longer require prophylactic regimens. We have a high-dose cohort, which is using 4 times higher dose at 2 times e to the 13th, which we announced recently that we just started dosing. That cohort will also have 5 patients, and we look forward to enrolling that over the coming months and presenting data, full data on the first cohort, which will likely include 6 months of follow-up at an academic conference in the second quarter, and hopefully having a year follow-up on the first cohort and perhaps 6 months follow-up on the second cohort, either at the end of this year or in early 2017. The one other thing that I'll just mention is the neutralizing antibodies. So we've now screened over 20 patients for neutralizing antibodies, pre-existing neutralizing antibodies to AV5, and only 1 of those patients tested screen positive and very marginally, and we think that's an area of differentiation that'll be important going forward.
Great. In terms of the goals and objectives before you move into phase 3, what do you feel like you need to show? And then as you do move into the next stages, this isn't really your traditional factor replacement approach, and so it may not be your traditional phase 3 hemophilia program. So maybe help us think about the next steps and what phase 3 might look like.
Sure. So I think what we want to, this is a dose-ranging study for a purpose. The purpose is to understand the relationship between the dose and the clinical outcomes and obviously the safety. So we are encouraged by the data that we saw. I think there's even an argument to be made that the low-dose cohort could have commercial viability, but we're very interested to see if we can generate, as we did in preclinical studies in non-human primates, an increase of factor IX expression at higher doses without any adverse safety profiles. So I think that's one of the things that we're interested in seeing. And then obviously understanding the durability of the factor IX expression over time. I think once we have an idea and we analyze all that information, we'll have our discussions with the regulators and we'll determine which dose to take forward into a pivotal study. And in terms of the pivotal study, it's obviously hard to say. We haven't had any dialogue with the regulators, but I think if you look at the enzyme replacement or long-acting therapies, they're typically looking at a single-arm study where the patients serve as their own control, somewhere around 40 to 50 patients, looking at factor IX levels, bleeding episodes, et cetera. And that's one point of departure that we might be able to look at.
Do you have thoughts as to kind of the threshold clinical profile for approval? What % of patients need to achieve what % factor activity for a certain period of time? Because I think we are departing from the traditional factor replacement. So maybe you don't need every single patient to be able to reduce or eliminate prophylactic factors. So where do you think those bars are, both for approvability and for adoption?
Yeah, I mean, obviously that's the critical question, and it's hard to answer that without having robust dialogue with the regulators. I think the best way I can answer that is that it's going to be a risk-benefit equation that the regulators and I think the clinicians are going to be looking at. I think if you have a very safe therapy that has really no significant adverse events, and you can impact a significant portion of those patients and have a clinical benefit, I think you have a commercially viable product. I think if your safety profile is all over the place and you've got a lack of predictability in the clinical outcome, I think it would be a tougher way to hoe, for lack of better words. So I think these are questions, obviously, that we look forward to answering as we have more data and begin to have discussions with the regulators.
And maybe talk about your access to the AAV5 vector, where it comes from, what your freedom to operate is, and/or exclusivity to the rights to the vector.
Sure. So we've licensed the AAV5 vector from the NIH. So we have exclusivity of using AAV5 in the area of gene therapy for liver-directed administration, as well as for the brain as well, or the CNS, more broadly speaking. We have exclusivity from the NIH, and those patents run into the 2019, 2020 range, depending on which territory that you're operating. We have an extraordinary amount of familiarity with AAV5. We've also developed our own mutations or variants of AAV5 that we've developed in-house. And we've also have a collaboration with 4D Molecular Therapeutics, which is actually, I think, presenting right across right now, where we are developing synthetic optimized vectors that will have greater tropism or affinity or optimization of transduction efficiency, targeting the brain and the liver. We have an exclusive partnership with them. We've selected those variants that we're going to be taking into preclinical validation studies this year, and hopefully we'll have some lead candidates that we'll be using by the end of the year.
Maybe take us through the Sanfilippo B program, what you've shown there, and when that's expected to move to the next stage of development.
Sure. So for those that aren't aware, Sanfilippo B is a lysosomal storage disease. There's probably about 2,000 patients that we believe suffer from Sanfilippo B in the major reimbursable markets. It's also called MPS IIIB. These patients are born without the ability to produce an enzyme called NAGLU. And NAGLU is used to metabolize a toxic substance that, if it builds up in the brain, can lead to severe cognitive decline and ultimately death by their teenage years. There's currently no therapy for these children, and it's obviously a devastating disease. In 2013, we provided and collaborated with the Institut Pasteur in France, where we provided them clinical material that we manufactured using our insect cell baculovirus expression system using AAV5. They administered that gene therapy in 4 children between 20 months and 53 months of age. In September, we announced, or our principal investigator announced, 12-month follow-up data that was very encouraging. And what we showed is that at 1, 3, and 12 months post-administration, that these patients had 14 to 17% expression of NAGLU from a baseline of 0, and that was detected in the CSF. We also examined clinical and cognitive endpoints through 3 different cognitive scales, and all patients during that year follow-up were acquiring skills. And then lastly, we looked at MRIs, which showed very preliminary evidence that there was no progression of brain atrophy. So we're very excited by that. We expect to take over sponsorship of that study shortly. There's an extension protocol that's being put in place where we're going to be looking at 30-month follow-up for these same 4 patients. So you're going to have the oldest child that'll be nearly 7 years of age. At that point in time, you would expect those children to be really crashing cognitively. And the youngest patient who was actually had a sibling who was with this disease and wasn't really manifesting symptoms will be nearly a little bit more than 4 years of age. So it'll be very interesting to see this data. The last patient will be evaluated in November of this year. We'll be aggregating the data and hopefully presenting it early in 2017.
Great. I think we're at a time, a strong cash position, over $200 million, a great partnership with Bristol-Myers focused on heart failure and beyond, and looking forward to more updates. Thanks so much, Matt.
Thanks a lot, Josh. Thank you.